So good morning, everybody. Thank you for taking the time to listen to my presentation. It's a little awkward for me to speak to a computer and not see the audience. So I try to do my best, and so I can't really respond to your reactions because I don't see you. But please type the questions, and then at the end of my presentation, I try to answer as many as I can. All right, so I'm going to talk a little bit about the microbial degradation of chlorinated pollutants, focused on chlorinated pollutants, and then a little bit about the principles of degradation and also some aspects of applied work. So now the next slide is a table, and this is a table that lists the priority pollutants that are listed on the ATSDR, the Agency of Toxic Substances and Disease Registry website. So that's from 2011, and it's a ranking of the top 100 pollutants. And if you go through this list, and now on the next slide, slide number 3, I counted all of these, the chlorinated pollutants that are listed on this site. And more than half, 56 of the top 100 pollutants listed are actually chlorinated compounds. So the point is that chlorinated chemicals are major environmental pollutants. And so that's why we are so interested in knowing and learning more about the microbiology contributing to their degradation. The next slide, it talks about sources of chlorinated contaminants. And clearly, human activities are a major source of these chlorinated chemicals that cause problems and concerns in the environment. So we also talk about anthropogenic activities. That's Greek. Anthropos means people, genesis, creation. So anthropogenic contaminants generated by people. And then you can imagine we have intentional and unintentional releases of these chemicals. I'll give you a few examples of intentional and unintentional release as I go through my talk. Then another term that has been coined based on this anthropogenic generation of these chlorinated chemicals is the term xenobiotic. So xenos, Greek for foreign, and bios, life. So the idea was that these chlorinated chemicals are foreign to life. And so we come back to that because that requires a little bit more discussion. On the next slide, with several pictures, I show you an example of a chlorinated chemical that was regarded as a poster child chemical for a xenobiotic. We talk about polychlorinated dibenzodioxins. In the center top, I show this general structure. So you have two benzene rings that are substituted with chlorine substituents. And these two benzene rings are bridged with two oxygens. So that's why we also call these chemicals, I mean, it's kind of a jargon abbreviation. We call them dioxins. But it would be more correct to talk about PCDDs, polychlorinated dibenzodioxins. So dioxin became infamous in Europe because of an accident in 1976 called the Seveso accident, and dioxin was also called at the time the Seveso poison. You see on the left, it had tremendous effect on the environment. It was an accident in a chemical company, and I'll show you exactly what happened on the next slide. But dioxins also were part of Agent Orange that was used as a defoliant, for instance, in the Vietnam War. And the consequences of that you can still see today in the bottom right. On the top right, you may remember the president of Ukraine about 10 years ago. He was poisoned in an attack, and you see the effects, the immediate effects of dioxins. You get a disease called chloracne, and your skin reacts, what is very obvious in this picture. So the point I want to make that polychlorinated dibenzodioxins were viewed as typical xenobiotics, man-made compounds. So briefly about this Seveso accident. So the idea was to use a chemical reaction to generate 2,4,5-trichlorophenol based on a chlorinated benzene, and at a temperature of 158 degrees, that would give you the desired products, in this case a herbicide, 2,4,5-T, and then also hexachlorophene, a disinfectant and fungicide. What happened there was that the reactor overheated and the temperature was higher. And then you generated in this reactor what was called the Seveso poison, specifically 2,3,7,8 tetrachlorodibenzodioxin. So this was an unintentional production. It was an accident. The reactor exploded and released about 1 kilogram of this toxic material. So in the last 10 to 20 years, we learned a lot about chlorinated chemicals in the environment. And it was initially surprising that there are natural processes that can contribute to the formation of chlorinated chemicals. So for example, combustion processes like forest fires, volcanic activity, but also chemical processes in the subsurface, photochemical processes, and even biological processes can generate halogenated, including chlorinated chemicals. Now coming back to this poster child xenobiotic, these polychlorinated dibenzodioxins and also polychlorinated dibenzofuranes shown in the structure, in the lower structure. So we have this unintentional production. Undesired byproducts in chemical synthesis, what I showed you. Then also incinerators generate these chemicals. But now, to some surprise, forest fires, geologic formation, so chemical reactions, and also biological reactions contribute to the formation of these chemicals. And if you look at the numbers, they're actually quite significant. Just forest fires produce about 60 kilograms of this material per year. In this accident in Italy, only about 1 kilogram was released. So the point here is that this term xenobiotic, foreign to life, may be misleading, because even chemicals that we viewed as anthropogenic, made by humans, are actually found in nature. And this is a very important observation. If you go to the next slide. Oops. I expand a little bit on this. This is from a publication that was published in Environmental Science and Technology. There's a long list of chemicals that were measured in fumes or gases released from volcanoes. And I highlighted in red, we talk a little bit more about this later, a few chemicals that you probably know or recognize as typical groundwater pollutants, specifically tetrachloroethene, trichloroethene, all dichloroethene isomers, and even vinyl chloride, chloroethene, were all detected in emissions from volcanoes. So clearly, there is a natural source of these chemicals. I go even a little further, specifically talking about vinyl chloride. And this has been documented in different publications, different approaches. In soil, you have organic matter that can be degraded to catechol. And in the presence of iron, you can form benzoquinone. And again, in the presence of ferric iron and oxygen and inorganic chloride, you can, under abiotic reactions, generate chlorinated organic compounds, including vinyl chloride. Now, of course, the concentrations are very low, but the point is that there are naturally occurring processes that generate chlorinated compounds, some of which are known as priority pollutants. Now, on the next slide, it shows you the number of known natural organohalogens. So compounds that carry a halogen bound to a carbon atom. And in 1954, we only knew 12, very few. So in 2010, this number has increased significantly to nearly 5,000 compounds that we recognize are produced naturally and have carbon halogen bonds. And the vast majority of those compounds are actually chlorinated compounds. So the conclusion here is that if there's natural production of these chemicals, there must be pathways and mechanisms that degrade these chemicals. If that wouldn't be the case, the oceans would be full of halogenated or chlorinated chemicals. But that's obviously not the case because nature has invented pathways and mechanisms to degrade these compounds. And this is the basis of bioremediation. So we're trying to understand what pathways, what mechanisms, what organisms exist that we can then benefit from by exploiting their activities in bioremediation. So then on the next slide, I give you a little bit history view of biodegradation of chlorinated chemicals. And I think it's maybe not the first compound that has been looked at in more detail, but it was probably a starting point. It comes back to the use of herbicides, insecticides like DDT. And I show the structure here of DDT. The slide is entitled Discovery of a Problem. So DDT was a solution for many problems at the time. It was a fantastic chemical for combating insecticides that would harm high-value crops. It was also fantastic insecticide to control mosquito populations and malaria. And you see the application in the bottom left part of that slide. So it was used extensively. Now, people then recognized that there may be problems associated with the use of DDT. And that's shown in the right-hand side of the slide. In the graph on the top, people measured the thickness of eggshells from predatory birds. And starting with the use of DDT to the end of DDT use, you can see that the eggshells were significantly thinner. And at the bottom, you see the consequences. When the bird tries to sit on the eggs, they would break. So the use of DDT had a tremendous impact on bird populations and actually also other predatory or at the end of the food chain organisms, including humans. So when this was discovered, DDT was banned and degradation of DDT was studied. And those were probably the first degradation studies on the next slide. So in the early 60s, focused on DDT, degradation of a chlorinated chemical. And since then, obviously, a lot of work was accomplished. Many different chlorinated pollutants have been investigated, and organisms have been identified, pathways and mechanisms, how these chemicals can be degraded. On the next slide, I show you the principal mechanisms, how microbes can break carbon halogen bonds. So what you see on the left side is an organic compound. The X symbolizes a halogen. So it could be a iodine, a bromine, a chlorine, or a fluorine substituent. Obviously, we are focused on chlorinated chemicals, but what I'm saying here for chlorinated chemicals also applies to halogenated chemicals in general. So now this carbon-halogen bond can be broken in different ways. First of all, you can replace it with a hydroxyl group, OH. That's shown on top. The OH group can be, or the oxygen in the OH group can be derived from oxygen. Then we call this oxygenolytic dehalogenation. The oxygen can also be derived from water. Then we call it hydrolytic dehalogenation. So this is a big difference because obviously when you have oxygenolytic dehalogenation, you require oxygen. Hydrolytic mechanisms can also function in the absence of oxygen. So that's important to remember. You also have a third possibility that's shown at the bottom, where the halogen substituent is replaced with a hydrogen. This is called reductive dechlorination. It typically occurs only under anoxic conditions. And what you need in order to perform reductive dechlorination, you need an electron donor, ED. So you need an electron donor to accomplish this type of reaction. So also note in the middle of the slide, in the center, that the halogen substituent is always released as an anion. So in the case of chlorine, you generate chloride, just like sodium chloride, salt. So this halogen substituent is always released as an anion, which is environmentally of no concern. What you also note is that a proton is released together with the chloride. So if you have a lot of dechlorination, you may acidify the environment because you do generate a proton. And this can be significant and can be important for bioremediation. We may talk again about this later. So now to the next slide. This is some general considerations and shows you the relationship between carbon oxidation state and the potential for oxidative or reductive transformation reactions. So on the left-hand side, you see an arrow, and it says carbon oxidation state. So the carbon oxidation state is an important factor determining if a compound can be oxidized or reduced. So the maximum reduced state of carbon is -4. The maximum oxidized state is +4. So you see that there's a significant difference in oxidation state between methane and carbon tetrachloride. The same is true for ethene and PCE, shown in the middle, or from benzene and hexachlorobenzene, shown on the right. So the potential for oxidative transformation increases as the chlorine substitution, the degree of substitution with chlorine atoms decreases. So benzene is very easily oxidized. Hexachlorobenzene is not. Now the potential for reductive transformation increases as the degree of chlorination increases. In other words, hexachlorobenzene or PCE are more susceptible to reductive transformation than they are to oxidative transformation. But this is important to keep in mind. Basically says if you have organic compounds with a high degree of chlorination, the likelihood that they undergo reductive transformation in anoxic environments is high. The probability that they are transformed in the presence of oxygen is relatively low. This is again illustrated in the next slide, where you see the relative trends of oxidative and reductive transformation, and here also shown sorption and volatilization rates. So on the x-axis, on the left-hand side, you have very few degree, a low degree of chlorination. On the right-hand side, you have polychlorinated chemicals. So if you have these polychlorinated chemicals, they have a high tendency to undergo reductive dechlorination, but they also have a tendency to sorption. Obviously, the size of the molecule, in other words, how many carbons you have in your compound of interest, also play a role here. If you have on the left-hand side monochlorinated chemicals, they are much more likely to undergo oxidation, and it's much more likely that they have a tendency to volatilize. On the next slide, I show you that this has a significant effect for the environmental fate of these chemicals. So shown here, it says biomagnification. It has to do with these more highly chlorinated compounds that have several carbon atoms, like PCBs, polychlorinated biphenyls, the PCDDs we discussed before. The problem here is that you may have very low concentrations in the water, but then when microbes, zooplankton, ingests these chemicals, and then small fish eat the zooplankton, the small fish are eaten by larger fish, and the predatory birds eat the larger fish, or humans get these fish. You can see with each tropic level, you concentrate these chemicals, and by the time the end consumer at the top of the food chain eats the food, they can accumulate very high concentrations of these chemicals, and that has uh, consequences. And this is known as biomagnification. And chemicals that have many chlorine substituents and are larger molecular size chemicals have a tendency to biomagnify. In contrast, on the next slide, if you have chemicals with lower degree of chlorination and also only a few carbon atoms, they have a tendency to volatilize. And that can have other consequences shown here. You have contamination. The groundwater moves these chemicals, because they are soluble to some degree, to residential areas, and you have a problem that is known as vapor intrusion. This will not happen with polychlorinated biphenyls because they sorb and are not volatile. But these smaller chemicals with lower degree of chlorination, they can volatilize and cause issues known as vapor intrusion. So now, when you think about bioremediation on the next slide, slide number 20, there are three key questions. And this was already in an NRC report discussed back in 1993. But these three questions are still valid. The key question is the chemical of concern degradable? I'm going to talk a little bit about that. And I give you examples for these different processes I mentioned before: oxygenolytic, hydrolytic, and reductive dechlorination. The slide here shows an interesting organism that is growing with 1,10-dichlorodecane. Dichlorodecane is misspelled in the title, I apologize. So on the left-hand slide, you see a tube. The bottom part is the medium, and the upper layer is 1,10-dichlorodecane. It's basically not soluble in water. But when you inoculate with this Pseudomonas strain, it grows vigorously, and the 1,10-dichlorodecane is completely degraded. And that's shown in the top right part. You see in the filled circles, 1,10-dichlorodecane is consumed. Chloride is released, stoichiometric amounts of chloride in the filled triangles, and the organism is growing. In the next slide, you see how this works. So you see 1,10-dichlorodecaine. You need oxygen, and the chlorine is cleaved off and replaced with an oxygen. And then the aldehyde group is oxidized to a carboxylic group. You basically have a fatty acid, which is degraded by a well-known mechanism called beta oxidation. That's how the organism gets carbon and energy. What I want to point out is that if you look at oxygen highlighted in red, only one of the oxygen atoms is actually incorporated into the product, this aldehyde. The other oxygen atom from oxygen goes into H2O. And so this mechanism is catalyzed by an enzyme system called monooxygenase. So such monooxygenases can be important for oxygenolytic dechlorination. On the next slide, I have another compound called 2-chlorobenzoate. There are many organisms that can degrade 2-chlorobenzoate with oxygen. If you look here, the oxygen is used and both atoms of the oxygen are incorporated into the product. The product is actually catechol, and these two on the right side, and these two hydroxyl groups, the oxygens, are derived from oxygen. So both oxygen atoms are incorporated into the product. Those enzymes are called dioxygenases. And so there are many dioxygenases known that can contribute to the dechlorination of chlorinated aromatic and aliphatic compounds. Another example for oxygenolytic dehalogenation. Then an example for hydrolytic dechlorination. In this case, hydrolytic dechlorination of 1,2-dichloroethane. Several organisms can do this. The best studied organism is Xanthobacter. So you have 1,2-dichloroethane, and the product you can find is 1,2-dichloroethanol, which the organism degrades further to CO2. So you see the chlorine is removed, replaced with a hydroxyl group. Now the question was, where is it coming from, the oxygen? Is it coming from oxygen or is it coming from water? And so people did studies with labeled water, H218O, and the label was found in 1,2-dichloroethanol. Pretty good evidence that the oxygen in the hydroxyl group in 1,2-dichloroethanol is derived from water. So that's an example for a hydrolytic dechlorination mechanism. This mechanism can function in the absence of oxygen. So for example, under anoxic conditions. But then the further degradation of 1,2-dichloroethanol in this particular organism, Xanthobacter, does require oxygen. So the overall process can only occur under aerobic conditions. On the next slide, I have an example for reductive dechlorination. And so this was first described back in '82, but it took a long time before people realized the significance of this finding. At the time, people did experiments with 3-chlorobenzoate, and that was dechlorinated, reductively dechlorinated, to benzoate. And what was interesting is that this reaction shown here, this reductive dechlorination reaction, was sufficient to sustain growth of this organism desulfomonylate TGI. So this was a very curious finding. And it took a few years, at least 10 or 15 years, to actually recognize the relevance of this process for bioremediation. On the next slide, this reductive dechlorination process linked to energy gain is now called organohalide respiration. In principle, you need a chlorinated compound, an electron donor. The chlorinated compound is reduced, you generate HCl, and this is an energetically favorable process. So in this little cartoon, you see the microbe is happy, it's smiling, because it has an electron donor available, and it has a chlorinated compound available. The chlorinated compound is reduced, and in this reduction, energy is released, which the organism can capture in the form of ATP. In other words, it can grow based on the energy it gains from reductive dechlorination. And those organisms are called organohalide respiring organisms. The process is called organohalide respiration. On the next slide shows you the application of this process to the reductive dechlorination of chlorinated ethenes, PC, TC, dichloroethenes, and vinyl chloride to non-toxic ethene. So each step requires an electron donor. The next slide, you can see that each reductive dechlorination step generates energy the organism can capture, and this is called organohalide respiration. So there are some other terms in literature like chlororespiration, dechlororespiration, chloridogenesis, metabolic reductive dechlorination. I suggest don't use those terms anymore. Try to use this term organohalide respiration because that's now used in the literature or should be used. On the next slide, you see a few of the populations involved in this process. So there are populations that only dechlorinate PC to TCE, like Desulfitobacterium. Then there are some organisms that dechlorinate PCE to cis-DCE. Some are listed here, but then they stop. What's critically important is that the reductive dechlorination is complete, going to ethene. And shown in the next slide is the reason why. Because dichloroethenes and vinyl chloride are still toxic. And actually the MCL, the maximum concentration limit set by the EPA, is only 2 ppb for vinyl chloride, so lower than for the other polychlorinated ethenes. So the question was, how can microbiology catalyze the last two reductive dechlorination steps? And in the next slide, we found or shown is the solution. A few dedicated microbiologists in different laboratories have found Dihalococoides, and that's the keystone bacterium responsible for these last two dechlorination steps. Dihalococoides. I'm going to talk a little bit more about those guys. I want to point out on this slide that this first reductive dechlorination step, or both steps from PC to TC to cis-DCE, those are actually catalyzed by a number of organisms. And you can, in many environments, assume that PCE is dechlorinated to cis-DCE if the conditions are conducive for microbial activity. That's not the case for the latter steps, catalyzed by Dihalococoides. I'll go back to that in a second. So then just briefly, this is a new paper that is just being published now. And I know that some of you have finally learned how to pronounce Dihalococoides, what wasn't easy. And then initially, Steve Zinder called the isolate he obtained Dihalococoides ethanogenous. But ethanogenous was misleading, and we renamed this organism. So it's now called Dihalococoides mccarthyii, in honor of Perry McCarthy, who was such a leader in this research, leading actually to the discovery of Dihalococoides. So in the next slide, you see a tree. And what we've done is that all of the known Dihalococoides strains all belong to Dihalococoides mccarthyii. So the term Dihalococoides ethanogenous should no longer be used. Okay, so all known strains of Dihalococoides belong to the species mccarthyii. And you see in this tree, and I have this green box around it, there's this class Dihalococcoidea. And there are other organisms that are not called Dihalococoides, like Dihalogenimonas or Dihalobium. So there are other organisms in this class, Dihalococcoidea, that are not Dihalococoides mccarthyii. And we are just now learning and understanding the diversity of this group. And it's very likely that more organisms will be found belonging to these other types of Dihalococcoidea, including Dihalogenimonas and Dihalobium. Okay, On the next slide, I want to talk now a little bit more about bioremediation and putting the Helicoccoides to work. Before I do this, I have a few definitions about bioremediation and also monitored natural attenuation. And this may be clear to most of you. I just want to make sure we are on the same page. So in bioremediation, you use biological systems, in this case microbes, to reclaim soil, sediments, and waters polluted by chlorinated toxic chemicals that are a threat to human and environmental health. The options you have include biostimulation, so you manipulate site conditions to increase the rate or extent of chlorinated compound degradation, or bioaugmentation, so you introduce organisms that can dechlorinate and degrade the target compounds. Monitored natural attenuation is a different approach because you don't manipulate the environment, you rely on a variety of physical, chemical, and biological processes that act without human intervention to achieve site-specific remediation objectives within reasonable time frames. And that's the EPA definition. And the person who was heavily involved in doing this work is Scott Wilson at the EPA. So the next slide. Oh, I'm in trouble here. Okay, next slide. I show you. No, okay, I misspoke. I mean John Wilson, okay, not Scott Wilson. John Wilson, EPA 1999. Okay, here, bioreactive barrier technology. This just gives you an example how biostimulation and bioaugmentation can work. So you see a cartoon of a plume. In red, this is the contaminant plume. You can intersect this plume and you can add stimulants that make native, so naturally occurring microbes in this environment more active, organisms that degrade the chlorinated compound of interest, and you generate an active zone, and the water that's leaving this active zone ideally has no concentrations of the chlorinated pollutant. If biostimulation alone is insufficient, the organisms may not be present or not be present in numbers high enough, you can consider bioaugmentation to accelerate the rates. And this is shown on the next slide in one of the first pilot studies where a lot of data were collected to demonstrate the value of biostimulation and biostimulation combined with bioaugmentation to treat a plume of predominantly dichloroethene, cis-dichloroethene. What's shown here is just the layout of these plots where the injection wells, the monitoring wells, the extraction wells were located. The key point is that the plots were adjacent to each other and they were hydraulically not connected. So what was done in one plot did not affect the adjacent plot. The next slide shows the results. In a control plot, the contaminant cis-dichloroethene was not removed. Very little happened. In the plot that was amended with lactate as an electron donor, you see that after about 6 weeks, ethene was formed and cis-dichloroethene was dechlorinated. On the right-hand side, you see the bioaugmentation plot. And you see here that when we added organisms, in this case, a mixed culture containing D. helicoccoides, we got ethene, but the time it required for cis-dichloroethene degradation to ethene was much shorter. So biostimulation alone was sufficient, but the rates were much higher when we did bioaugmentation combined with biostimulation. So that's a nice example, and this technology has been applied to many, many sites and is often successful. So what is typically done at sites, you can monitor the D. helicoccoides population. And this is shown on top. So this big white circle indicates the total bacteria, and in blue, the D. helicoccoides. And you can see that D. helicoccoides makes up a significant proportion of the total bacteria. Before we did this treatment, the D. helicoccoides were below 0.001% of the total bacteria. So this treatment significantly increased the number of D. helicoccoides, and that is associated with the observed dechlorination rates. Now you can get a little bit more specific. There are different types of D. helicoccoides, different strains, and they have different reductive dehalogenases. So they do different things in this pathway from PC to ethene. And this is shown on the bottom. So you have this red proportion, that's a gene encoding a reductive dehalogenase that dechlorinates TC and cis DC to vinyl chloride, but doesn't dechlorinate vinyl chloride to ethene. Initially, this was very abundant. But then as more and more cis DC was formed in vinyl chloride, you can see that other reductive dehalogenases become more abundant. In yellow, for example, VCRA, in green, BVCA. So we looked at this type of analysis at many sites. And this may be of interest to some of you. How can we use this type of information to make predictions if ethene is formed? And so what we found is, that's at the bottom, you can, for example, using molecular tools, determine the total number of D. helicoccoides and estimate the total number of bacteria based on 16S rRNA genes. And if the ratio of D. helicoccoides to total bacteria is about 0.05%, it's very likely that ethene is formed. Also, when you see D. helicoccoides 16S rRNA genes at abundances exceeding 10 to the 6 per liter, it's very likely that ethene is formed at chlorinated ethene contaminated sites. Also, if the D. helicoccoides 16S rRNA gene to VCRA and BVCA ratios are near unity, it's also very likely that you see ethene formation. Now go to the next slide. It's a cartoon. It's a cartoon about bioaugmentation. And you can see the idea is Pseudomonas pudita from the laboratory is brought back into the environment. And these guys are so spoiled, adapted to the conditions in the laboratory. They don't want to go back outside and compete with all these rough guys. It's kind of a joke, but there's some truth to it. So when you have organisms, you take them from the environment, you have them in the laboratory, and you're nice to them, and then you bring them back into the environment, they don't perform. They lost the ability to compete with other organisms in the real environment. But this is apparently not the case for D. helicoccoides. D. helicoccoides has a different behavior. And this is very important. So what's so special about D. helicoccoides on the next slide? I'll just point out a few things that make D. helicoccoides very special. In contrast to many other microbes, it has a very small, streamlined genome. So it's very small. In other words, it can't lose anything. If it loses genes, it can't grow at all. If you take a Pseudomonas that have a much larger genome, it has many genes it can lose when you cultivate it in the laboratory. And it will do that. And then you bring it back out to the real environment, and it doesn't have these genes anymore, and it can't compete. It's one of the problems that apparently cannot happen with D. helicoccoides. Another important observation is that D. helicoccoides has a very specialized lifestyle. It can only grow with hydrogen as electron donor and a chlorinated compound as electron acceptor. It can't do anything else. So when you actually have these contaminants out there and there's hydrogen flux, these organisms will do the job because they can't do anything else. With many other organisms, they can dechlorinate a chlorinated compound, but they have also other possibilities to generate energy with other substrates. So now you have to convince them to use the chlorinated compound, but they may not do because they have other options. Another thing is when you have Pseudomonas without food, or many other organisms, they don't survive very long. So we have experiments where we didn't feed the helicoccoides for many months or even years. If you provide them with chlorinated substrate and hydrogen, they recover very quickly and do the job. So another unique feature. And then they're also very competitive for hydrogen. So very low hydrogen concentrations they can utilize. I see I have to talk a little faster. So on the next slide, I want to expand a little bit on other chlorinated chemicals. So for example, 1,2-dichloropropane. Misspelled in the title, I apologize. So 1,2-dichloropropane is another chlorinated solvent, was frequently used in agricultural purposes, is often found also as a groundwater contaminant. So we found the helicoccoides can also dechlorinate 1,2-dichloropropane, but typically not the diheliococcoides that can dechlorinate chlorinated ethenes. Those are specific strains of Diheliococcoides mccartyi. And also Dehalogenimonas can dechlorinate 1,2-dichloropropane. And those organisms possess a unique gene that we call DCPA, dichloropropane reductive dehalogenase, that we identified in these specific Diheliococcoides strains and Dehalogenimonas. And this can be a target now to see if these organisms, these types of organisms, are present or not. And so molecular tools to detect DCPA are available now, and MI has access to that. So can I talk 5 minutes more maybe? Okay, so then another class of compounds of interest are chlorinated methanes. And so illustrated here, carbon tet, chloroform, dichloromethane, chloromethane, and then methane. I'm not implying that this pathway works like this, but just an analogy to the chlorinated ethenes. So we know that DCM and chloromethane are easily degraded under oxic conditions, but we don't really know much about the anoxic degradation. We go to the next slide. This is a collaboration with Elizabeth Edwards at the University of Toronto. They found a culture that can reductively dechlorinate via organohalide respiration chloroform to dichloromethane. So chloroform to DCM. Unfortunately, DCM was not degraded further, and it actually inhibited chloroform degradation to DCM. So we must find solutions to get rid of DCM. And so typically we would go to contaminated sites to find organisms that can degrade DCM. In this case, we had a different strategy. So we went to a place that is not contaminated, at least not the place where we went. It was actually a very pleasant environment, a rainforest in Puerto Rico. We took samples there from the Rio Mameyes, and we got an enrichment culture that degraded DCM. It's shown on the next slide. So you see in red, DCM is degraded. When we spike DCM, it's degraded faster. The only product we find is acetate, shown in gray, and a type of Dihilobacter strain, shown in black, is growing when DCM is degraded. The bottom line is we had a culture that can degrade DCM. So now the idea is to combine on the next slide these two cultures, culture CF50 that dechlorinates chloroform to DCM, and the culture RM that degrades dichloromethane to acetate and inorganic chloride. So we got samples from a contaminated site that's shown in the next slide that was mainly contaminated with carbon TET. But a bioreactive barrier was installed based on zero-valent iron. The zero-valent iron dechlorinated the carbon TET to predominantly chloroform and DCM. We got samples from that site. In the next slide, we tried to do bioaugmentation in the laboratory. It's a laboratory-based study where we bioaugmented this material with culture CF50. And you see in red that chloroform was degraded, DCM was formed in blue. The respiked chloroform was degraded quickly, DCM was formed. We then added the DCM degrading culture RM. And somewhat disappointing, not much happened initially. But we knew from our laboratory studies before that this culture requires bicarbonate. So then we added bicarbonate because apparently this groundwater did not have a lot of bicarbonate. And after bicarbonate addition, DCM was degraded. When we respiked it, it was degraded very quickly. So the point here is you need to understand the microbiology in order to make bioaugmentation work. And it's not unreasonable to have 10 millimolar bicarbonate. Actually, many aquifers have that much bicarbonate. What we also noticed there is that carbon TET, CT, carbon tetrachloride, was very inhibitory to this process for both of these cultures. So carbon TET cannot be present to make this work. And on the next slide, this is the next question on the three key question slide. Is the environment habitable? And in this particular case, carbon TET would be a compound that would prevent these organisms from showing activity. So in order to make this environment habitable, some other mechanism must be used to remove carbon TET. In this case, a zero-valent iron wall took care and removed the majority of the carbon TET. So now the last question here. What is the rate limiting factor and can it be modified? So if the rate limiting factor on the next slide is electron donor, if you want to stimulate reductive dechlorination processes, you can add electron donor by biostimulation to increase hydrogen flux and support the key dechlorinators with the electron donor they require. Maybe the organisms are not present. It is possible that you work at sites and dihalococoides are simply not there or present in very low abundances. You can do bioaugmentation. To do bioaugmentation, on the next slide, cultures are commercially available. These are two of the ones that I am familiar with, KB1 and SDC9. I'm sure there are others. The next slide, just briefly, dihalococoides are picky eaters. And so what they need is hydrogen. So whenever you do biostimulation, what you actually try to do is to increase the flux of hydrogen. And I mentioned before that dihalococoides are very competitive hydrogen utilizers. So the idea of adding a lot to have a lot of hydrogen may not necessarily help dihalococoides. It helps a lot other organisms that cannot dechlorinate, but not necessarily dihalococoides. So you have to strike a balance here between increasing hydrogen flux, but not overloading the system with other negative effects, like secondary effects that degrade groundwater quality. So there's a lot of research going on at the moment to actually demonstrate how you measure if dihalococoides is hydrogen limited, indicating you need to add more electron donor. But it's not needed to add more electron donor as long as these organisms are not hydrogen limited. Another thing they need is B12. For reductive dechlorination, they need a corinoid B12 enzyme system. The problem is they can't make B12. what is kind of curious. So they require other organisms to generate the B12. And there are many different types of B12 out there. And the chemistry is rather complicated, so I'm not going into this. The point I want to make is that there's also a lot of research going on right now to figure out what partner populations can actually provide the B12 dihelicocoides can use, because many of the B12 or chlorinoid molecules other organisms make, dihelicocoides cannot use. So that's a very interesting question. So then on the next slide, a few general thoughts about moving science into practice. So if you want to do bioremediation, active treatment, biostimulation, maybe combined with bioaugmentation, you have to pay for it. It's expensive, costs money. So maybe monitored natural attenuation will do the job just as fine, and it's cheaper. But maybe another technology is even more appropriate than bioremediation or M&A. Now, you can talk to many people, you get different opinions. So you can be pointed in different directions, right? And probably many people have a point, but it's just difficult to see the entire picture, what's really going on at your site. So the danger, of investing in a technology that in the end is not as productive as another technology is rather high. So what we need to do, we need to come up with a set of tools that would facilitate decision making. On the next slide, so we need tools to support meaningful and site-specific decision making with the goal for efficient implementation of the most promising remedial strategy. And so tools that we are developing and that are available are these molecular biological tools and also compound-specific isotope analysis. And you can learn a little bit more about these tools if you go to the ITRC Environmental Molecular Diagnostics Team website. And I indicated the URL of the ITRC on this slide. So you can also just Google ITRC EMD team and you will find this. So yeah, you can learn a little bit more about these new tools and what they can do. So the cartoon kind of shows you what we want to do. We need to get more information out of a subsurface environment. The problem is we can't see it. So we need tools that help us to understand what's going on. So then on the next slide, just briefly, on the left-hand side, you see traditional site data that have been collected for many years. And I'm not suggesting that we should stop collecting this type of information. But what is pretty clear to me and many others is that this type of information is insufficient to make good decisions at all sites. So what we need to do is we need to also collect these MBT, compound-specific isotope analysis information, develop a multiple lines of evidence approach for what remedy is most promising, and then make remedial actions. If you don't have all this information together, you're guessing. Maybe you're lucky and you implement the right strategy, but maybe you're not. Then on the next slide, just briefly, very briefly about these molecular biological tools. When I talk about this, I typically mean quantitative real-time PCR, qPCR approaches that are very sensitive and give you quantitative information about organisms like dihelicocoides that are tightly linked to a process, reductive dechlorination. But we also can target process-specific biomarkers. Example here also genes of dihelicocoides, BVCA, VCRA are genes that encode specific reductive dehalogenases involved in ethene formation. On the next slide, lots of colors. different compounds, chlorinated ethanes, chlorinated ethenes, chlorinated methanes, chlorinated propanes. In gray are organism-specific biomarkers that are available. And in green are process-specific biomarkers that target reductive dehalogenases involved in these different dechlorination steps. I think the point I want to make is that we are getting better and better developing understanding of these specific biomarkers. We can go to the environment, take a sample, and then ask specific questions. Are these biomarker genes there? Are they active? How abundant are they? And we can give information about possible and actual activity. Next slide. I think we are coming to the end. I'm a little late, a little slow. What I show on the left are near to midterm accomplishments or vision. And some of these items I've listed, we already accomplished. So we know a lot more about the microbiology. We know a lot about biomarkers. We can certainly identify more, but we already know a lot. The technology to detect and quantify them is improving. We are developing standardized procedures so that different laboratories can easily standardize their SOPs, their standard operating procedures, and different laboratories will most likely come up with the same result for the same sample. We can tailor the analysis to specific sites so that not all of these targets must be analyzed at all sites. We develop understanding of the evolutionary and adaptive mechanisms, how D. helicoccoides, for example, acquires the ability to efficiently dechlorinate vinyl chloride. Hopefully, we can use these tools to develop understanding of rate constants. That would allow us to make meaningful data interpretation and informed decision making. With the ultimate goal to transition bioremediation from a trial and error practice to a scientific approach with predictable outcomes. The next slide is the final slide, and I just want to acknowledge many people in my group. Also, longtime collaborator Elizabeth Edwards at the University of Toronto, Kurt Pinnell, Tufts University, and as we do more and more genomic work, Kostas Konstantinidis, a good colleague at Georgia Tech. And then obviously I have to acknowledge a number of people working for the Navy and also many consultants who supplied us with ideas and also sample materials from many different contaminated sites. And then the funding for this work is predominantly provided by the Department of Defense, SERDAP, ESTCP, and some funding for the chlorinated methane work was provided by DuPont. And with that, the last slide I have is the big question mark. And I think that's the opportunity for you now to ask questions. And I will try and do my best to answer them as good as I can. Thank you.
Thank you, Frank, for a very interesting, informative presentation. So now we'll go and try to answer a few of the questions. How will the helicoccoides do in high levels or free product environments?
So we did experiments in source zones, and we have very sophisticated experimental setups with one-dimensional continuous flow columns and also two-dimensional boxes. That is work that is done in collaboration with my colleague Kurt Pinnell and Natalie Capiro at Tufts University. What we find is the following. In source zones with high concentrations of PCE and TCE, D. helicoccoides don't do very well. But that is not terribly surprising because other organisms, for example Geobacter lovelii and other PCE to cis-DC dechlorinators, are much better at dechlorinating PCE and TCE to cis-DCE. They do it faster than Dihalococoides can do. So in source zones with high concentrations of PCE, other organisms take over, but they only dechlorinate to cis-DCE. The cis-DCE will move away from the source. And under those conditions, Dihalococoides is favored. So the cis-DC leaving the source then is dechlorinated by Dihalococoides in some distance from the source to vinyl chloride and ethene. What may be important to recognize is that some of these organisms that dechlorinate PC to cis-DCE do it so fast that the dechlorination rate, as long as electron donors provided, the dechlorination rates of PCE exceed or can exceed the dissolution rate of PCE into the aqueous phase. So we have systems in the laboratory where we have a DNAPL present. You can see it. But the aqueous phase PCE concentration is 0 because the organisms dechlorinate the PCE faster to cis-DCE than the PCE can go into solution from the NAPL. But that's not done by Dihalococoides. The cis-DC that's generated by this process, downstream of the source, that's where Dihalococoides comes into play.
Are there bacterial strains that break down chloroform?
So chloroform, that's what I mentioned in my talk. So Elizabeth Edwards, she has a paper, one of her students in 2010, where they describe a mixed culture that contains a Dihalobacter strain that can respire with chloroform as electron acceptor. The product is dichloromethane. So the dichloromethane is not transformed further. There are other laboratories that have similar cultures. They dechlorinate, these cultures dechlorinate chloroform to dichloromethane. The known cultures all contain a type of Dihalobacter strain responsible for chloroform to dichloromethane reductive dechlorination. What I described in my talk is a separate culture that was derived from this pristine environment in Puerto Rico. We have an enrichment culture called culture RM. That culture cannot dechlorinate chloroform, but it can degrade dichloromethane. We have combined the chloroform dechlorinating culture with this dichloromethane degrading culture and have now a consortium that can actually completely degrade chloroform to non-toxic products, including acetate and inorganic chloride.
Okay, another question. Electron donors cause a decrease in pH at most sites. What is the practical limit for effective biodegradation to continue by Dihalococoides McCartyi?
Good question. So especially when you have a source zone, you can generate a significant amount of chloride. That means you generate a lot of hydrochloric acid. So the pH may go down. And Dihalococoides is somewhat sensitive to pH fluctuations. Well, maybe I shouldn't say fluctuations, but it doesn't perform well in low pH environments. So most of the experiments we do in a laboratory are at circumneutral pH. I would say that's between 6.8 and 7.5. Dihalococoides performs very well. When the pH goes below 6.5, the rates slow down. They slow down even more if you go down below to pH 6. And at pH 5.5, they don't do anything. If you keep them at pH 5.5 for a certain amount of time, you increase the pH again, they recover. But we have not been able to really find Dihalococoides activity at pH 5.5 and below. And we also noticed when the exposure time to low pH environments exceeds a certain amount of time, you do lose activity and it doesn't rebound. So pH is of concern. And I would certainly be worried if the pH drops below 6 that you lose activity based on Dihalococoides. Now, there are ways of buffering a subsurface environment, but that's always complicated. I think you also refer on your question, let me see, uh, when you add electron donor, so it's the pH decrease is not really caused by the dechlorination per se, by formation of hydrochloric acid. It can also be caused by electron donor addition because you add substrates that can be fermented, you generate acid. And that can contribute to a drop in pH. And that's what I briefly mentioned. Simply adding a lot of electron donor may do more harm than benefit just because of pH decreases. So we have to be smart about electron donor additions. We need to make additions when they are needed to avoid pH drops and also to save money. And there are approaches to add electron donor that may work better than others. So this is an area of research what I'm particularly interested in. I would like to find biomarkers that tell us when Dihalococoides is hydrogen limited. If we can accomplish this goal, we could tell people now the rate is limited by hydrogen availability, hydrogen flux. You have to increase it. Then you can add electron donor. But as long as hydrogen flux is sufficient for Dihalococoides, no additional electron donors needed. And in my view, we add too much. So it would be great to have a tool to quantify that and give people, the green light. Now you need more electron donor. So we are working on this.
Okay, one last question. What are the bacteria capable of 1,2-DCA degradation, and what would be representative concentrations in cells per milliliter to indicate biodegradation is occurring?
So 1,2-DCA, 1,2-dichloroethane. So many Dihalococoides that dechlorinate chlorinated ethenes like cis-DC, vinyl chloride can also degrade 1,2-dichloroethane. But there are many other organisms that can also degrade 1,2-dichloroethane. I personally have limited experience with 1,2-dichloroethane contaminated sites. So I would not be able to give you good numbers. How many cells per mil or per liter are, can be used as an indicator for 1,2-DCA degradation. My assumption would be that it's not very different from what we use for predicting vinyl chloride to ethene transformation. So when I told you before, if you see 10 to the 6 Dihalococoides cells in a chlorinated ethene contaminated environment, it's very likely that you see ethene formation. If you have a 1,2-dichloroethane plume and no other contaminants, only 1,2-dichloroethane, and you see an abundance of Dihalococoides in the range of 10 to the 5 to 10 to the 6 or even higher per liter, I would argue this is a very strong indicator that the microbiology is working and transforming 1,2-DCA to ethene. But again, so my experience with 1,2-DCA is somewhat limited. So I can't give you a better answer than what I just tried to do.
Okay, so unfortunately, we're out of time. For any additional questions, we will try to email you responses to those questions, or you can also contact us if you send us an email at info@microbe.com, microbe.com, then we will get with Dr. Loeffler and make sure we get answers for you on those questions. Again, we appreciate you joining us today and hope that you'll be available to join us on future webinars. Thank you.
Thank you.